Okay. All right, light reactions. Okay, let's get our frame of reference one more time here. So we're looking at a thylakoid here. Okay, so here is a chloroplast with many chloroplasts in one cell. And if you look at one chloroplast and zoom in on this stack here, this is a thylakoid, another thylakoid, another thylakoid, a stack of thylakoids. It's called a granum. Very good. So there's one thylakoid. So this is the fossil bilayer thylakoid membrane. In the membrane, we have a number of structures. Embedded, we have photosystem 2, photosystem 1, what we'll call a proton pump, some electron carriers, and an enzyme called Nadp Reductase. Okay? So we're going to look at the process of light reactions, and by the end of today, hopefully, if we don't run out of time, you should understand what's happening, what's the purpose of the light, what's the purpose of electron carriers. Proton gradient is a key term to know. And we should see examples of active and passive transport, which we have already learned. Okay, so let's label some structures first. So here we have photosystem 2. Oh, I'm having some. Oh, I came out of blood.
Cut. Take 3, Mr. Allen.
I gotta recalibrate. Press pause there for me.
Alright, we're gonna cut.
Good. And we're back. Okay, so we have photo system 2, and I'll be lazy here. This is photo system 1. All right, and the reason why we have 2 before 1 is their name in terms of when they evolved. So early photosynthetic bacteria had a photosystem. This one evolved later in eukaryotic cells.
I never got that drawing of the thing to label.
Okay, here we go, bud.
That's your fault, Sam.
Yes, Sam did do it.
Alrighty, so photons are little packages of light, and so these little lightning bolts represent photons. Photon. And we know that in the visible light spectrum, there's different sorts of photons, colors of the rainbow, variety of diff. So this is a proton pump, and as the name suggests, it's going to pump protons from what concentration to what concentration?
Low to high.
Low to high, yeah, that's what a pump does. High-low would just be facilitated diffusion. This is going to be a type of active transport.
So it's low to high?
Low to high, yeah. So I'm going to put little stars on these things. These are electron carriers. All they're going to do is carry electrons from A to B. So I'll put a little star here. Electron carrier. And this structure is called Nadp Reductase. That sounds scary.
Is that
Sorry?
something we need to memorize?
Yeah. So to reduce. When something becomes reduced, it seems back-asswards, but it's to gain electrons. Okay? Now, why would it be gaining electrons if it's reduced? The reason we call it reduction is when a molecule gains electrons, its stability is reduced. It becomes more reactive.
What's it called again?
This NADP reductase. I'll write it more neatly over here, bigger so you can see it in the back. NADP reductase. Okay, and A's, who remembers what that means? Anything ends with A, like lactase,
Lactose.
breaks down lactose.
A process.
Breaks down.
It's an enzyme that's going to catalyze or kickstart a chemical reaction. So this is an enzyme that reduces NADP. So it's going to give electrons to NADP to reduce it. Okay? Oh, and this structure is really important. Oh, ATP, we've heard that before. Usable form of energy by the cell. ATP synthase. Okay? So the name tells you what it does. Ase means it's a
Active.
Ase enzyme. It's an enzyme. It's going to synthesize what? ATP. Okay? The name tells you what it does. Okay, let's start looking at the process. So, photons of light. We'll start here at this end of the electron transport chain. Why is it called that? Because it's a pathway, a physical pathway that electrons go through. Okay, so we have a photon hitting photosystem 2, and that's going to excite electrons. Specifically, remember there's a bunch of accessory pigments, and when they get hit by the photon of light, they vibrate in place, and they channel that vibrational energy or kinetic movement energy to the next structure, and eventually gets to that special spot called the reaction center. And the reaction center has a special type of chlorophyll, and when it receives the energy, it doesn't vibrate, but what does it do? Right, it gives the energy to a couple of electrons, and when those electrons become energized, they're very, very, very unstable, and they're easily removed by an electron carrier. So they're kicked out, basically. Okay? So this yellow pathway represents the movement of electrons. When the chlorophyll in the reaction center loses electrons, they have to be replaced. They can't keep doing that. Right? So, water.
So they're replaced by splitting H2O.
Right, we're going to split water, and that's going to be our source of electrons. So right here we have water. Now, if we take two waters, how many oxygens in two H2Os?
2.
2. So we get molecular oxygen produced, and we get 4 hydrogen ions and 4 electrons. Okay? And the hydrogen ions from the water are going to be in the lumen or thylakoid space. Thylakoid, lumen, thylakoid space, same thing. Okay? That's a bit small from where you're sitting. So we take 2 waters, okay, and we're going to split it up. We're going to make an O2, because 2 H2Os give us 2 oxygens, and 4 electrons, and 4 hydrogen ions. All this was once water, but we split it apart. And the protons are going to stay for now. They're going to stay in the thylakoid lumen, or thylakoid space, adding to what's referred to as a proton gradient, which is just a concentration gradient of protons. So we're building up a concentration of protons here. That's going to be really important later to power up ATP synthase. Okay? The electrons, where do you think those are going to go?
Back into the photon thingy, not the photon.
Yeah, in the photosystem. So the electrons that have been lost by chlorophyll are replaced by the electrons that we achieve from splitting of water. Okay? And if we look at your equation, the summary equation for photosynthesis, we use carbon dioxide, and we use water, and we use energy from the sun, and that's going to yield us C6H12O6, which is their sugar. It's the whole point of photosynthesis. And we're going to make some oxygen. Okay, so already we've seen why we need water for a source of electrons and the oxygen that's produced from splitting water.
So is C6H12O6, is that ATP?
That is glucose. That's the sugar.
glucose?
So the whole point of making, of going through photosynthesis is so the plant can make its own food, which is sugar. And it can be used to make glucose, a type of sugar. ATP, we'll see in a second here. Okay, so far, back up in a second, photon hits the reaction center. That energy is going to be used to eject some electrons from the chlorophyll in the reaction center, and the electrons are picked up by an electron carrier. Meanwhile, there's an enzyme, it has a very unique name, enzyme Z. I don't know where they get that name. I guess they ran out of creativity. And the enzyme will split the water. Why? To replace?
To replace the electrons that were lost.
Exactly, replace those electrons that were lost. And in the process, we produce a waste product, oxygen, which is why plants produce oxygen, which we're going to need ourselves. Anything that's going to use oxygen, we have plants to thank for our supply. And the protons, the H's, from the splitting of water collect in the thylakoid space right here. Okay, so this electron carrier is going to pick up electrons from photosystem 2, specifically the chlorophyll in the reaction center of that photosystem, and they take them over here, the proton pump. So we'll try to keep this kind of simple, but anytime you have a watery environment, it could be your glass of water, your coffee, your apple juice, the extracellular environment of a cell, or the this would be the cytosol of this cell because we're still inside the cell, or this would be the stroma of the chloroplast. Anyway, anytime you have a bunch of water, some of the waters are split up into H's and OH's, specifically hydrogen ions and hydroxide ions. So, there are a bunch of hydrogen ions scattered through here. When this pump, working with this electron carrier, receives the electrons, it's going to pull hydrogen ions, or protons, same thing, going to pull them from the stroma into the thylakoid space, helping create the what? Proton gradient, or concentration gradient of protons. So we have two sources of protons here. The first one we saw was from splitting of water, yeah. And the second source is by pulling them from the stroma into the inside of the thylakoid, or thylakoid space. So you have this big concentration of protons, a proton gradient. Okay? So let's park that for a minute. We're going to use that shortly. So as electrons get passed from one structure to the next, they lose energy. It's like passing a hot potato around. It's really hot, and you gotta pass the next person, and it warms my hands, it warms Sam's hands, and so on. And eventually, the potato cools off, right? It loses energy. Same kind of thing happens to electrons as they go from here to here to here to here. They've lost energy. And they can't really do any work for us anymore. By work, I mean create a proton gradient. So electrons are going to be passed into this photosystem, which had previously ejected electrons, just as what happened over here. Okay? So these low energy electrons replace the ones that were previously lost. What happens here? Another photon of light hits this photosystem 1, excites those electrons, re-energizes them, and they become very, very stable or unstable? Unstable, good. So this thing here, called a what?
Proton carrier.
Electron carrier.
Close electron carrier, yeah. And these all have names, but we won't clutter our brains with paradox and plastoquinone. It's just too complicated. We'll call them all electron carriers. So these electrons are high in energy. They're not going to be used to create a proton gradient like over here, but they're going to be passed on to Nadp Reductase. And that enzyme is going to take NADP plus electrons, right, plus some hydrogen ions, which are found out here, and we're going to make NADPH. So, NADPH is essential to drive the dark reactions, which we'll call the Calvin cycle. And the Calvin cycle is where we actually make sugars. So we haven't seen where we're going to use carbon dioxide yet. We haven't seen sugar yet. Okay? Now, sorry?
So you said NADPH is essential for the dark cycle?
Right, so there's two things the Calvin cycle needs. We need a supply of carbon, we need NADPH, and we need ATP. We haven't made ATP yet, right? But if you remember, a few minutes ago, I was talking about a particular structure that's going to do this. What's it called?
Mitochondria?
That's a different process, we'll say later. What was it?
The ATP synthase.
Right, ATP synthase is going to synthesize or put together ATP, okay? Remember the proton gradient? Okay, so in nature, we don't like, we like equilibrium, things that are the same. We talked about diffusion, concentrations of molecules in a spread out. So here we have a high concentration, but these are ions, these protons. They can't go through the phospholipid bilayer. We know that. But they can go from high to low concentration through some kind of protein, right? That would be an example of what kind of transport? Not active, it's going to be passive. What kind of passive? Facilitated diffusion, good. Because it needs the help of the membrane protein to get through. Okay? But this is not just a channel protein, a hole going through the membrane. This is an enzyme. So, this is kind of cool. As protons flow from high concentration to low concentration, it physically makes this enzyme spin incredibly fast. And in the stroma, which is a fluid outside the thylakoid, we are going to take ADP. Do you remember how many phosphates ATP has? 3, because it's a adenosine triphosphate. So what do you think ADP has? 2. So there's a bunch of ADP. Let me write that a bit neater. There's a bunch of ADP floating around in the stroma. There's also a bunch of phosphates. Well, let's do some advanced math here. ADP plus one more P can give us
ADP 2.
ATP, right? So 2 plus 1 is 3. So this product is going to be ATP, which is also used in Calvin cycle. So basically what this enzyme, ATP synthase, does is it's going to, so I'm ATP synthase, okay? I have ADP because it has 2 phosphates, all right? So as protons flow through ATP synthase, it spins around, and as it does, it changes shape, much like the carrier proteins change shape when the solute molecule attaches to it. So by changing shape, it can grab onto ADP and a P. And as it changes shape, as it spins, it can kind of put these really close together, and then they bond together, and now I get ATP, and we let go of it into the stroma. And by the way, it's in the stroma where we will have Calvin cycle occur. So, we've made 2 products. The electrons that originally came from water go through here become part of NADPH. The ATP produced by the proton gradient are going to be used in Calvin cycle, as is NADPH. Lucas? Go ahead.
We
Yeah.
didn't get to that.
You say?
The Calvin cycle is the dark cycle, right?
Yeah, Calvin cycle, dark reactions, same thing.
Does the thylakoid take the ADP and P from the stroma?
Yeah, there's a bunch of stuff that's in the stroma. We have protons, they're just there. We have ADP, they're just there. We have phosphates, they're there. We have ATP, we have NADPH. There's a whole bunch of things, raw materials needed to make these 2 substances. Okay, let's do a quick recap from the beginning. Photon hits photosystem 2. That energizes electrons, become very unstable. They're picked up by an electron carrier. Let's rewind. Meanwhile, those electrons that were lost must be replaced. So there's an enzyme called Z, enzyme Z. It splits water. When we split water, if you take 2 water molecules, so for every 2 waters we split, we get 2 O's. They go on to form what's called molecular oxygen. You've heard that before, O2. Okay? The hydrogens, the protons and electrons become disassociated from each other. The electrons are going to go into here, replace those lost. The protons they're gonna be kind of dumped into the lumen, the thylakoid lumen, thylakoid space, whatever. Adding to the proton gradient, which we'll use in a minute. Okay, so meanwhile, here's our electron carrier. It takes electrons over to the pump. The pump is going to kind of think that electrons are negative, protons are positive, and love in chemistry, opposites attract. And so it kind of pulls protons through the membrane, through that protein there. And the protons get moved into the thylakoid space, adding to the proton gradient. Okay. And now what can we say about the energy level of the electrons? They've been kind of used to pull some protons in. Every time the electrons go from one structure to another, they lose energy. So now this electron carrier is carrying what is very stable electrons. And it gives into this photosystem, which had previously ejected electrons. So these ones have been replaced. This is photon of light, channel the energy down in the reaction center, boom, energize electrons, eject them. Another electron carrier picks them up. High or low energy of electrons right now? High, because they just got ejected, right? And so we have this electron carrier takes them to this enzyme with a big ugly name, Nadp Reductase. A's means it's an enzyme. It's going to reduce, which means something's going to gain electrons. And what's going to gain electrons? NADP, okay, which is floating around in here. So this enzyme is going to reduce or give electrons to NADP. Where are electrons coming from? Through the electron transport chain, okay? And so when we get these NADP plus some electrons plus proton, we get NADPH. That's using Calvin cycle. Meanwhile, this concentration of gradient of protons, or proton gradient, they flow through ATP synthase. It spins, grabs on ADP and P, or phosphate, and ADP plus P gives us ATP. And there's our second product of that required for Calvin cycle. Students always get this mixed up. The protons flowing through here do not become part of ATP. They flow through there, and then maybe they're pumped back in. They flow through there, and then maybe they're used to make NADPH. They don't become part of ATP. The only thing that is used to make ATP, the physical supply structures, are ADP and P. Okay, so next would be the Calvin cycle. I'll give you some review to do on that first before I move on. All right, cut.